define more genetic alterations unique to a tumor that could be exploited for new therapies. Researchers now use RNAi and genome editing technologies to identify genes that, when inactivated, cause tumor cells but not normal cells to die. This approach is for identifying synthetic lethal interactions between different genetic alterations that on their own are not lethal was pioneered in budding yeast with the help of development of genome-wide small hairpin RNAs, shRNAs, libraries, collections of RNAi constructs that target every gene in the human genome, and other genome editing methodologies such as the CRISPR-Cas9 system. This approach is now also feasible in human cells. Tumor cells and normal cells are infected with pools of shRNA constructs, each of which harbors a unique sequence tag known as a barcode. After a period of growth, the RNA constructs can be isolated with shRNAs that were lost from the pool can be identified by sequencing of the remaining constructs. The shRNAs that were lost point to the target gene being essential for viability in the cell type from they were lost. Those shRNAs that were lost point to the target gene being essential for viability in the cell type from which they were lost. Those shRNA constructs that cause lethality in tumor cells but not normal cells suggest that the genes that target are essential for the survival of a tumor cell but not a normal cell. This approach has been used, for example, to identify genes that, when inactivated, cause selective lethality in cancer cells harboring an oncogenic form of RAS, the KRAS oncogene. The proteins encoded by these genes could provide novel targets for the development of new therapeutics for tumors that harbor oncogenic forms of RAS. The vision for the future of medicine is that modern sequencing technologies as well as genome-wide RNA and protein analysis technologies will allow doctors to classify a tumor and provide a comprehensive list of the oncogenic lesions that drive cancer growth. Treatment will then be tailored to be unique properties of each patient's cancer, and many cancers such as breast cancer and CML, we can already see this future taking shape. Then some key concepts: the genetic basis of cancer. Dominant gain of function mutations in proto-oncogenes and recessive loss of function mutations in tumor suppressor genes are oncogenic. Among the proteins encoded by proto-oncogenes and growth-promoting signaling proteins and their receptors, signal-transducing proteins, transcription factors, and semi-anti-apoptotic proteins. An activating mutation of one of the two alleles of a proto-oncogene converts it into an oncogene. This can occur by point mutation, gene amplification, gene translocation, or misexpression. Tumor suppressor genes encode proteins that directly or indirectly control progression through the cell cycle, such as checkpoint pathway proteins that arrest the cell cycle if a previous step has occurred incorrectly, components of growth inhibiting signaling pathways, and pro-apoptotic proteins. The first tumor suppressor gene to be recognized, RB, is mutated in retinoblastoma and many other tumors. Some components of the RB pathway is altered in most tumors. Inheritance of a single mutant allele of RB greatly increases the probability that a specific kind of cancer will develop, as is the case for many other tumor suppressor genes like APC and BRCA1. In individuals born heterozygous for a tumor suppressor gene mutation, a somatic cell can undergo loss of heterozygosity or LOH by mutation or a deletion of the normal allele, chromosome missegregation, mitotic recombination, or gene silencing. Mutations affecting epigenetic regulations such as histone modifying enzymes or chromosome remodelers are associated with a variety of tumors. Then microRNAs can promote or inhibit tumorigenesis by affecting the expression of multiple oncoproteins. Novel sequencing technologies have greatly accelerated the discovery of genes involved in cancer and are having a profound impact on cancer diagnosis and treatment. The advent of molecular techniques for characterizing individual tumors is allowing the application of drug and antibody treatments that target the properties of a particular tumor. This strategy permits more effective treatment of individual patients and reduce the use of drugs or antibodies that will be ineffective and possibly toxic. These refinements have allowed substantial reduction in breast cancer mortality. Novel shRNA and genome editing methods allow for the identification of genes specifically required for the survival of cancer cells, thereby facilitating the discovery of new therapeutic targets. Then misregulation of cell growth and death pathways in cancer. In this section, we examine in more detail how the deregulation of growth promoting and growth inhibiting signaling pathways contribute to tumorigenesis. We first discuss how mutations that result in the unregulated constitutive activity of certain proteins or in their overproduction promote cell proliferation and transformation. Next, we discuss how loss of function mutations in differentiation pathways contribute to tumorigenesis. We end this section with a description of how misregulation of genes that control programmed cell death, such as p53, drives tumorigenesis. The oncogenic receptors can promote proliferation in the absence of external growth factors. Hyperactivation of a growth-inducing signaling protein due to an alteration of a protein might seem a like mechanism of cancer, but in fact, this rarely occurs. Only on such naturally occurring oncogene has been discovered, the cis-oncogene, which encodes an altered form of platelet-derived growth factor (PDGF), can aberrantly stimulate proliferation of cells that normally express the PDGF receptor when expressed at higher levels. A more common event is that cancer cells begin to produce an unaltered growth factor that acts on the cancer that produces it. This phenomenon is called autocrine stimulation. In contrast, oncogenes encoding cell surface receptors that transduce growth promoting signals have been associated with several types of cancer. Many of these receptors have intrinsic protein tyrosine kinase activity in their cytosolic domains, which is quiescent until activated. Ligand binding to the external domains of these receptor tyrosine kinase RTKs leads to their dimerization and activation of their kinase activity, initiating an intracellular signaling pathway that ultimately promotes proliferation. In some cases, a point mutation changes a normal RTK into one that dimerizes and is constitutively active even in the absence of ligand. For instance, a single point mutation converts the normal human EGF receptor into the NEU oncoprotein NEU for its first non-role in NEU neuroblastoma, which is an initiator of certain mouse cancers. Similarly, human tumors called multiple endocrine neoplasia type 2 produce a constitutively active dimeric glia-derived neurotrophic factor GDNF receptor that results from a point mutation in the extracellular domain. The GDNF receptor and the HER2 receptor are both protein tyrosine kinases, so the constitutively active forms excessively phosphorylate their downstream target proteins. In other cases, deletion of much of the extracellular ligand binding domain produces a constitutively active oncogenic receptor. For example, deletion of the extracellular domain of the normal EGF receptor converts it into the domestic ERBB oncoprotein from erythroblastoma virus, in which a viral version of the altered gene was first identified. Mutations leading to overproduction of a normal RTK can also be oncogenic. For instance, many human breast cancers overproduce a normal HER receptor. Here we can see a picture which represents effects of oncogenic mutations in proto-oncogenes that encode cell surface receptors. A mutation that alters a single amino acid valine to glutamine in the transmembrane region of HER2 receptor causes dimerization of the receptor even in the absence of a normal EGF-related ligand, transforming it into the oncoprotein NEU, a constitutively active kinase. Right, the deletion that causes loss of the extracellular ligand binding domain in the EGF receptor leads, for unknown reasons, to constitutive activation of the kinase activity of the resulting oncoprotein ERBB. Like here, proto-oncogene receptor proteins, HER2 receptor, EGF receptor, exterior cytosol, valine region, then inactive receptor tyrosine kinase is there, oncogenic mutation is deletion, NEU oncoprotein is there, exterior cytosol, glutamine, ERBB oncoprotein, constitutively active protein tyrosine kinase, etc. can be seen. Then, because of amplification of its encoding gene, as a result, the cells that are stimulated to proliferate in the presence of a very low concentration of EGF are related hormones. Concentration is too low to stimulate proliferation of normal cells. Many oncogenes encode constitutively active signal transducing proteins. A large number of oncogenes are derived from proto-oncogenes whose Encoded proteins are components or regulators of signal transduction pathways, most prominent among them in the RAS pathway. RAS is a key component in the transduction of signals from activated receptors to the cascade of protein kinase. In the first part of this pathway, a signal from an activated RTK is carried via two adapter proteins to RAS, converting into the active GTP bound form. In the second part of the pathway, activated RAS transmits the signal via two intermediate protein kinase to MAP kinase. The activated MAP kinase then polymerates a number of transcription factors that include synthesis of important growth and proliferation proteins. Virtually every component of this RTK RAS MAP kinase signaling cascade has been identified as an oncogene or tumor suppressor gene. Among the best studied oncogenes are the RAS-D gene themselves, which were the first non-viral oncogenes to be recognized. Growth hormone, tumor suppressor gene, RTK, RAS MAP kinase pathway components are frequently mutated in cancer. Components of the RTK RAS MAP kinase pathway in which oncogene mutations have been identified in human cancers are highlighted in green. When we look at the picture, we could see growth hormone is attached to the receptor. Proto-oncogenes are represented by green. Tumor suppressor genes are represented by red color. So RAS is their GDP bound form, but tumor suppressor gene is associated with that. Then N-terminal regulatory domain is there. MEK, MAP kinase, transcription factor, transcription, etc. can be seen. Any one of a number of changes in RAS can lead to its uncontrolled and therefore dominant activity. In particular, if a point mutation substitutes any other amino acid for the glycine at position 12 in the RAS sequence, the normal protein is converted into a constitutively active oncoprotein. The simple mutation reduces the protein's GTPase activity, thus maintaining RAS in the active GTP bound state. Activating RAS mutations short circuit the first part of the RTK pathway, making upstream activation triggered by ligand binding to the receptor unnecessary. Constitutively active RAS oncoproteins are produced by many types of human tumors, including bladder, colon, mammary, skin and lung carcinomas, neuromas, and leukemias. Constitutive RAS activation can also arise from the recessive loss of function mutation in a GTPase activity protein, GAP. The normal function of GAP is to accelerate hydrolysis of GTP and thus the conversion of active GTP bound RAS to inactive GDP bound RAS. The loss of GAP leads to sustained RAS activation of downstream signal transducing proteins. For example, neurofibrinomas, a benign tumor of the sheath cells that surround nerves, is caused by loss of both alleles of NF1, which encodes a RAS GAP type protein. Individuals with neurofibromatosis have inherited a single mutant NF1 allele. Subsequent somatic mutations in the other allele leads to formation of neurofibromas. This NF1, like RB, is a tumor suppressor gene, and the tendency to develop neurofibromas like hereditary retinoblastoma is inherited as an autosomal dominant trait. Oncogenes encoding other altered components of the RTK RAS MAP kinase pathway have also been identified. For example, constitutively active forms of RAF have been identified in approximately 50% of melanomas. As in the case of constitutively active forms of RAS, this mutant RAF forms are no longer responsive to regulatory signals coming from the cell surface and signal continuous cell growth and proliferation. In addition to RTK, RAS MAP kinase signaling pathway constituents, cytoplasmic protein kinases are frequently mutated in cancer. Instead of cell first oncogene to be discovered, VSRC of Rous sarcoma retrovirus encodes a constitutively active protein tyrosine kinase. At least 8 mammalian proto-oncogenes encode a family of non-receptor tyrosine kinase related to the SRC protein. In other instances, kinases are fused to other proteins and doing the protein kinase with new specificity. The BCR-ABL fusion protein is an example of such an oncokinase. As described above, hyperactive kinases and oncokinases have been successfully targeted in cancer therapy. Then inappropriate production of nuclear transcription factors can induce transformation. Mutations that create oncogenes or damage tumor suppressor genes eventually cause change in gene expression. These changes can be measured by comparing the amounts of different mRNAs produced in normal cells and in tumor cells. Since the most direct effect on gene expression is exerted by transcription factors, it is not surprising that many oncogenes encode transcription factors. Two examples are JUN and FOS, which initially were identified in transforming retroviruses and later found to be overexpressed in some human tumors. The JUN and FOS proto-oncogenes encode proteins that sometimes associate to form a heterotrimeric transcription factor called AP1 that binds to a sequence found in promoters and enhancers of many genes. These proteins function as oncoproteins by activating the transcription of key genes that encode growth promoting proteins or by inhibiting the transcription of growth repressing genes. Many nuclear proto-oncogene proteins that produced when normal cells are stimulated to grow, indicating their direct role in growth control. For example, PDGF treatment of quiescent mouse 3T3 cells induce an approximately 50-fold increase in the production of the transcription factor FOS and MYC, the products of the FOS and MYC proto-oncogenes. Initially, there is a transient rise of FOS and later a more prolonged rise of MYC. The levels of both proteins decline within a few hours, a regulatory effect that may in normal cells help to avoid cancer. The FOS and MYC proteins stimulate transcription of genes encoding proteins that promote progression through the G1 phase of the cell cycle and G1 to S transition. In normal cells, FOS and MYC mRNAs and the proteins they encode are intrinsically unstable and degrade rapidly after the genes are transcribed. Some of the genetic changes that turn FOS from a normal gene into an oncogene involve deletions of the sequences that normally make the FOS mRNA and protein short-lived. Conversion of the MYC proto-oncogene into an oncogene can occur by different mechanisms. In cells of the human tumor known as Burkitt's lymphoma, the MYC gene is translocated to a site near the heavy chain antibody genes, which are normally active in antibody-producing white blood cells. Experiment figure: addition of serum to quiescent 3T3 cells leads to marked increase in the activity of two proto-oncogene products FOS and MYC. Serum contains factors such as platelet-derived growth factor (PDGF) that stimulate the growth of quiescent cells. One of the earliest effects of growth factor is to induce expression of FOS and MYC. Those encoded proteins are transcription factors. Then Burkitt's lymphoma, chromosomal translocation and Burkitt's lymphoma. As a result of translocation between chromosome 8 and 14, the MYC gene is placed adjacent to the gene 4 part of the antibody heavy chain, leading to overproduction of MYC transcription factor in lymphocytes and hence their growth into the lymphoma. The MYC translocation is a rare aberration of normal DNA rearrangements that occur during maturation of antibody-producing cells. The translocated MYC gene, now regulated by the antibody gene enhancer, is continuously highly expressed, causing the cell to become cancerous. Localized amplification of a segment of DNA containing the MYC gene that occurs in several human tumors also causes inappropriately high production of the otherwise normal MYC protein. The MYC gene encodes a basic helix loop helix protein that acts as part of a set of interacting proteins that can dimerize in various combinations, bind to DNA, and coordinately regulate the transcription of target genes. Other members of this protein set include MAD, MAX, and MNT. MAX can heterodimerize with MYC, MAD, and MNT. MYC MAX dimers regulate genes that control proliferation, such as cyclins. MAD proteins inhibit MYC proteins, which has led to an interest in using MAD proteins or drugs that stimulate MAD proteins to rein in excessive MYC activity that contribute to tumor formation. MYC protein complexes affect transcription by recruiting chromatin modifying complexes containing histone acetyl transferase, which usually stimulate transcription to MYC target genes. MAD and MNT work with the SIN3 co-repressor protein to bring in histone deacylase that help to block transcription. Together, all these proteins form a regulatory network that employs protein-protein association, variations in DNA binding, and transcriptional regulation to control cell proliferation. Overproduction of MYC protein tips the scales in favor of cell growth and division. Then aberrations in signaling pathways that control development are associated with many cancers. During normal development, secreted signals such as hedgehog, WNT, and TGF beta are used to direct cells to particular development fates, which may include the property of rapid mitosis. The effects of such signals must be regulated so that growth is limited to the right time and place. Among the mechanisms available for retaining in the effects of these powerful developmental signals are inducible intracellular antagonists, receptor blockers, and competing signals. Mutations that prevent such restraining mechanisms from operating are likely to be oncogenic, causing inappropriate or cancerous growth. The hedgehog signaling pathway, which is used repeatedly during development to control cell fates, is a good example of a signaling pathway implicated in cancer induction. In the skin and cerebellum, one of the human HH proteins, sonic hedgehog, stimulates cell division by binding to and inactivating a membrane protein called PATCHD1. Loss of function mutations in PTC1 permit cell proliferation in the absence of an HH signal. Thus, PTC1 is a tumor suppressor gene. People who inherit a single working copy of PTC1 have a property to develop skin and brain cancer. Loss of function mutations in PTC1 permit cell proliferation in the absence of an HH signal. This PTC1 is a tumor suppressor gene. People who inherit a single working copy of PTC1 have a propensity to develop skin and brain cancer. Either can occur when the remaining allele is damaged. Other people can get these diseases too if they lost both copies of the gene. Thus, there are both familial and sporadic cases of these diseases, just as in retinoblastoma. Mutations in the other genes in the HH signaling pathway are also associated with cancer. Some such mutations create oncogenes that turn on HH target genes inappropriately. Others are recessive mutations that affect negative regulators such as PTC1. As the case for a number of other tumor suppressor genes, complete loss of PTC1 function would lead to early fetal death. Since the protein is needed for development, so it is only the tumor cells that are homozygous. Many of the signaling pathways described in other chapters also play roles in controlling embryonic development and cell proliferation in adult tissues. In recent years, mutations affecting components of most of the signaling pathways have been linked to cancer. Indeed, once one gene in a developmental pathway has been linked to a type of human cancer, knowledge of that pathway gleaned from model organisms such as worms, flies, or mice allows focused investigations of the possible involvement of additional pathway genes in other cases of cancer. For example, APC, a gene that is mutated in the path to colon cancer, is now known to be part of the WNT signaling pathway. That knowledge in turn led to the discovery of beta-catenin mutations in colon cancer. Mutations in tumor suppressor developmental genes promote tumor formation in tissues where the affected gene normally helps restrain growth. Thus, mutations do not cause cancers in tissues where the primary role of the developmental regulator is to control cell fate, what type of cell develops, but not cell division. Mutations in developmental proto-oncogenes may induce tumor formation in tissues where an affected gene normally promotes cell proliferation, or in another tissue where a gene has become aberrantly active. Transforming growth factor beta, TGF beta, despite its name, inhibits proliferation of many cell types, including most epithelial and immune system cells. Binding of TGF beta to its receptor activates cytosolic SMAD transcription factors. After translocating to the nucleus, SMADs can promote expression of the gene encoding P15 and an inhibitor of cyclin-dependent kinase 4, CDK4, which causes cells to arrest in G1. TGF-beta signaling also promotes expressions of cell genes encoding extracellular matrix proteins and transmembrane activator inhibitor 1, which reduces the plasmin-catalyzed degradation of the matrix. Loss of function mutations in either TGF-beta receptors or in SMADs thus promote cell proliferation and probably contribute to the invasiveness and metastasis of tumor cells. Such mutations have in fact been found in a variety of human cancers. For example, deletions of the SMAD4 gene occurs in many human pancreatic cancers. Retinoblastoma and colon cancer cells lack functional TGF-beta receptors and therefore are unresponsive to TGF-beta growth inhibitors. Effect of loss of TGF-beta signaling. Binding of TGF-beta, an anti-growth factor, to its receptor causes activation of SMAD transcription factors. In the absence of effective TGF-beta signaling owing to either a receptor mutation or a SMAD mutation, cell proliferation and invasion of surrounding extracellular matrix increase. So TGF-beta is there. Loss of function mutation and receptors block TGF-beta signal. Type 2 receptors are there. Type 1 receptor is there. Then blocking is there. Cytosolic region, SMAD3, SMAD3B. Loss of function mutation and SMADs block TGF-beta signal. SMAD4, SMAD4. Then diseased production of P15 increases proliferation. Then P15 promoter, P15 nuclear region, then transcription of gene encoding cell cycle inhibitor is there. Decreased production of PAI1 allows increased extracellular matrix degradation and hence metastasis. Then PAI1 promoter, PAI1 transcription of gene encoding inhibitor of proteases that degrade extracellular matrix proteins. Then next topic, genes that regulate apoptosis can function as proto-oncogenes or tumor suppressor genes. During normal development, many cells are designed for programmed cell death, also known as apoptosis. Many abnormalities, including errors in mitosis, DNA damage, or an abnormal excess of cells not needed for development of a working organ, can trigger apoptosis. For stem cells, apoptosis appears to be the default situation and signals are required to ensure cell survival. In some cells, apoptosis appears to be the default situation and signals are required to ensure cell survival. Cells can receive instructions to live and instructions to die, and the complex regulatory system integrates these various kinds of information. If cells do not die when they should and instead keep proliferating, a tumor may form. For example, chronic lymphoblastic leukemia occurs because cells survive when they should not. The cells accumulate slowly and most are not actively dividing, but they do not die. CLL cells have chromosomal translocations that activate a gene called BCL1, BCL2, which we know how to be a critical blocker of apoptosis. The resultant inappropriate overproduction of BCL2 protein prevents normal apoptosis and allows survival of these tumor cells. CLL tumors are therefore attributable to a failure of cell death. Another dozen or so proto-oncogenes that are normally involved in negatively regulating apoptosis have been found to be mutated to become oncogenes. Overproduction of their encoded proteins prevents apoptosis even when it is needed to stop cancer cells from doing. Conversely, genes whose protein products stimulate apoptosis behave as tumor suppressors. An example is the PTEN gene. The phosphate encoded by this gene dephosphorylates phosphatidylinositol 3,4,5 triphosphate, a secondary messenger that functions in activation of AKT. Cells lacking PTEN phosphatase have elevated levels of phosphatidylinositol 3,4,5 triphosphate and active AKT, which promotes cell survival, growth, and proliferation and prevents apoptosis by several pathways. This PTEN acts as a pro-apoptotic tumor suppressor for decreasing the anti-apoptotic and proliferating promoting effects of AKT. The most common pro-apoptotic tumor suppressor gene implicated in human cancers is P53, pro-apoptotic tumor suppressor. Among the genes activated by P53 are several encoding pro-apoptotic proteins such as PAX. And when cells suffer extensive DNA damage or numerous other stresses such as hypoxia, the p53 induced expression of pro-apoptotic proteins leads to their quick demise. While apoptosis may seem like a drastic response to DNA damage, it prevents the proliferation of cells that are likely to accumulate multiple mutations. When p53 function is lost, apoptosis cannot be induced and the accumulation of mutations required for cancer to develop and progress becomes more likely. Then key concepts: misregulation of cell growth and death pathways in cancer. Mutations that permit receptors for growth factors to dimerize in the absence of their normal ligands lead to constitutive receptor activity. Overproduction of growth factor receptors can have the same effect and lead to abnormal cell proliferation. Most tumor cells produce constitutively active forms of one or more intracellular signal transducing proteins, causing growth promoting signals in the absence of normal growth factors. Inappropriate production of nuclear transcription factors such as FOS, JUN, and MYC can induce transformation. In Burkitt's lymphoma cells, MYC is translocated close to an antibody gene, leading to overproduction of MYC. Many genes that regulate normal developmental processes encode proteins that function in various signaling pathways. Their normal roles in regulating where and when growth occurs are reflected in the characteristics of tumors that arise when these genes are mutated. Loss of signaling by TGF beta, a negative growth regulator, promotes cell proliferation and development of malignancy. Overexpression of anti-apoptotic genes or loss of pro-apoptotic genes promotes tumorigenesis. The pro-apoptotic gene p53 is frequently mutated in cancers. Then deregulation of cancer cell and genome maintenance pathways in cancer, that we will be learning with the help of next video. Thank you for listening.